Welcome students to chapter 12 on the circulatory system. This chapter is one of the largest in the textbook, but fortunately for us, many of the concepts are lingering and or known to the students by common sense, which will help you proceed faster through this presentation. The chapter itself is divided into 6 topics, 6 sections. The first section is an overview of the circulatory system. Section B deals with the heart. Section C with the vascular system. Section D talks about the regulation of the circulatory system. And then section E focuses on pathophysiology. And the last section revisits homeostasis with respect to blood loss. Moving rapidly through these slides, the first thing to understand is that there are 3 principal components that make up the human circulatory system. There's a pump, which is the heart. There's the piping, which is the vascular system itself. And then the fluid that travels through this system, which is the blood. Whereas some components are under local control, most of the cardiovascular system is impacted by hormones from the endocrine system, information coming from the nervous system, and also the volume of blood and its composition can be regulated by the urinary system, which therefore also affects blood pressure and the pumping of the heart. The term Hematocrit refers to just the red blood cells present within blood, nothing else. If blood is taken from a patient and spun, the blood will settle into 3 layers. The heaviest layer will be the red blood cells. The middle layer will be the white blood cells and the platelets, known as the buffy coat. And the fluid component of the blood will be the plasma, which contains many dissolved materials, including nutrients and waste materials, and messages traveling throughout the body. Plasma itself is over 90% water, depending on the level of hydration of the patient. Plasma also acts as a conduit for transport and as a reservoir of chemicals in many cases. So nutrients, electrolytes, wastes, gases, hormones, and buffer proteins are all present in plasma. Serum is that component of the plasma when the clotting agents are removed. This includes fibrinogen and other proteins. All cellular components of the blood, as well as the platelets, are manufactured in bone marrow from stem cells, and the process is known as hematopoiesis. Thus, these stem cells are known as hematopoietic stem cells. Homeostatic feedback and local growth factors control the development down different paths for different types of cell products. By far the largest cellular component of blood are the red blood cells, known as erythrocytes. Their main function is to transport gases, dissolved gases. So they carry oxygen and carbon dioxide by way of a very high concentration of a protein called hemoglobin. The erythrocytes are a beautiful example of evolution. They have a biconcave shape, a small size, and they have a high surface to volume ratio. And that ratio increases the surface area, so maximizing the exchange of oxygen and carbon dioxide. In addition, to make more space for this hemoglobin, the mature erythrocytes, they have their nucleus removed and their organelles are either digested away or removed also. Since their capacity to regenerate has been eliminated, erythrocytes have a short half-life. They hang around in the human body for about 3 months, 120 days, before they are recycled in the spleen. Erythrocytes are synthesized, as we said earlier, in bone marrow, but there are 2 types of bone marrow, and the type of bone marrow that they are born in is called red bone marrow. And the process is called erythropoiesis. A hormone released by the kidney of all things, is then influential in dictating how many red blood cells are born in the red bone marrow. The hormone is erythropoietin. That is mentioned further in the chapter on renal function. The two major organs which take care of red blood cells, other than the bone, are the spleen and liver. As we mentioned a few minutes ago, the spleen removes old red blood cells, and it has a filtering effect where mature, healthy red blood cells are able to proceed on their circulatory journey. The liver metabolizes the byproducts from the breakdown of these red blood cells. That includes the iron, which is very precious to the body, and the iron is recycled by binding to various other factors that are present in the liver and the blood. Iron stores in the liver exist as iron bound to a protein called ferritin. If iron is needed by bone marrow, the liver solubilizes and releases the iron, but the iron has to be transported in the blood on another protein called transferrin, which delivers it to its target destination. With respect to metabolic activity, the human diet must contain iron, folic acid, and vitamin B12 for the system to function correctly in solubilizing and loading iron onto newly made red blood cells. Let's tackle a homeostatic mechanism that pertains to this topic. Assume that the delivery of oxygen decreases to the kidneys. The kidneys respond by releasing the hormone erythropoietin, which then travels through the blood plasma, and its target cells are in the bone marrow. The bone marrow responds by increasing the production of red blood cells, erythrocytes. So the level of blood hemoglobin increases, and the capacity to carry more blood increases because of the added red blood cells. And then that delivers a message back to the kidney by negative feedback that the concentration of oxygen is now back to healthy levels. Certain forms of kidney failure will affect the kidney's ability to produce erythropoietin. Therefore, in many dialysis patients where the same symptoms of kidney failure are present, these patients are given synthetic forms of erythropoietin to stimulate red blood cell formation in their bone marrow. This knowledge has become available to athletes who can then abuse these synthetic forms of erythropoietin to increase their blood content for carrying oxygen and therefore achieve an advantage over their competitors. However, the process is not without risk because the concentration of red blood cells does affect the blood in overall composition, making it too thick and viscous, and that can lead to strokes, clotting, and heart failure. Incidentally, testosterone enhances red blood cell production because it has a knock-on effect on the kidney in producing more of the erythropoietin. That means men who have high levels of testosterone should have high levels of red blood cells. Overall, men produce testosterone, therefore they have a higher hematocrit content than women. Anemia is a medical term applied to whenever a patient's oxygen carrying capacities are reduced. A multitude of reasons could exist, but some of the common ones are a decrease in the total number of red blood cells, or a decrease in the concentration of hemoglobin in each red blood cell, or a combination thereof. Sickle cell anemia, sickle cell disease, can also cause anemia because under certain conditions, especially low oxygen levels, the red blood cells in these individuals, which are genetically defective, begin to sickle, therefore their concentration reduces, and that reduces the ability to carry oxygen. Table 12.1 here lists the major causes of anemia in human populations. The first one is deficiency in the diet of one of the three commodities that we spoke about earlier, either iron, vitamin B12, or folic acid. Number two is a failure in the bone marrow due to cancer or toxic drugs. Number three is hemorrhage, not enough blood in the body. Number 4 is the lack of production of erythropoietin in kidney cells. And number 5 is the excessive destruction of erythrocytes in diseases like sickle cell anemia. Turning our attention next to the small percentage of white blood cells that are present in blood. These are known as leukocytes. And their main purpose is to assist the immune system in its function. Leukocytes can be divided into 2 groups based on morphological examination under the microscope. The first group are called granulocytes, because in their cytoplasm they contain granules. These are the neutrophils, the eosinophils, and the basophils, NEB for short. And then the second population are the agranulocytes. They do not contain distinct granules in their cytoplasm, and they are the monocytes, which give rise to the macrophages, and the lymphocytes, the B and T cells. Each cell population has a primary function within the blood. Neutrophils are phagocytes. That means they eat other things such as bacteria. An expansion in the number of neutrophils in a patient is indicative of infection. Likewise, eosinophils are increased in concentration when parasitic worms and such enter the body. These are eukaryotic parasites, and the granules contain toxic molecules which are released onto the surface of the parasites, hopefully in an attempt to control and kill them. Basophils are response cells. They monitor local conditions, and once triggered, they can release a whole host of different chemicals, amongst which is heparin, which is an anti-clotting factor, and that mops up any type of clot after an infection. And they can also release histamine, which is, as we know, a molecule that attracts other cells and can be a problem in allergies. Monocytes circulate in the blood and are phagocytic, and at the time of infection, they are able to leave the circulation and develop into macrophages in tissues and organs. Lymphocytes, as we'll talk about in another chapter on the immune system, are the major components of the adaptive immune system. Platelets are actually shreddings of cell cytoplasm, and these mother cells are located in bone marrow as megakaryocytes. So platelets are simply fragments broken away from these cells. They do contain granules within their cytoplasm, and those granules are important in forming blood clots. Platelets are therefore produced in bone marrow, and they exist in the circulation for a period of time. The pathway of development of hematopoietic stem cells is governed by a whole range of growth factors, some of which are listed here, and one of 2 which we have already discussed. Under normal circumstances, the circulatory system is a closed system. That means there should be no leaks as such. The heart is the epicenter of 2 circuits of this system. The systemic circuit that feeds the entire body, except the lungs, and the pulmonary system of the lungs. Blood vessels are divided into 5 categories: the familiar arteries, capillaries, and veins, but also the smaller arteries known as arterioles, and the smaller veins known as venules. The rule of thumb is that arteries are away, so arteries carry blood away from the heart, and all veins bring blood to the heart. Unfortunately, the use of oxygenated and deoxygenated blood does not work, because there are some arteries that carry both oxygenated and deoxygenated blood. The values in this table pertain to a human male at rest. So we can see that the majority of the blood is in the abdominal organs, with almost an equal amount in the skeletal muscles. The least amount of blood is actually present inside the heart. Interestingly, the total volume of blood that's moved is 5 liters per minute. The next section is both rewarding and frustrating for students simultaneously. We are going to look at some thermodynamics with respect to pressure, the flow of blood, and the resistance to that flow. A couple of definitions first. Pressure is the force exerted on the fluid to cause it to move. And in the case of blood, that pressure is measured in millimeters of mercury as a legacy of the past. Flow is the volume of blood that's moved in a particular amount of time, and in our case is measured in milliliters per minute. Therefore, there's an equation relating pressure to something called resistance. So resistance is the pushback that you get for blood flowing through any particular pipe. So resistance is measured as friction that impedes flow. The important equation, therefore, is flow is proportional to the pressure change, and flow is also inversely proportional to the amount of resistance. The analogy to a river or canal is always useful for students. In the case of pressure, that can be related to altitude. So a river that's steeper will have more water flowing than a river that's more level. Figure 12.7 presents two graphics to help us understand that. If the pressure is measured at point P1 inside the tube and also at point P2 and is assumed to be 100 milligrams of mercury here and only 10 milligrams of mercury there, then the difference between the 2 points is 90 milligrams of mercury. Therefore, the flow rate will be 10 milliliters per minute. 10 milliliters per minute. If the pressures are adjusted inside the same tubes to 500 milliliters of mercury at P1 and 410 millimeters of mercury at P2, the relative difference, the delta, is still 90 millimeters of mercury. And in this case, even though the pressures are much higher, the flow rate between the 2 points will still be 10 milliliters of fluid per minute. In this case, because the tubes are the same, the resistance will be the same. Thus, the equation flow is going to be proportional to the pressure. And because the pressure is the same, therefore the flow will be the same. As far as blood flow is concerned, there are 3 determinants of resistance. One is the thickness of the blood, known as viscosity. The higher the viscosity, the thicker the fluid. The second is the length of the blood vessel through which the blood is traveling. And the last one is the most important, is the radius of the blood vessel. And that radius can be controlled at local level as well as at the general level by increasing or decreasing the diameter of the blood vessels. The equation that relates all these terms together is Passui's law, a French scientist who worked out this relationship. Basically, if you double the radius of a tube, you will increase the flow of fluid 16 times. A compound view of the circulatory system with respect to the chambers and parts is given here. So the heart has atria and ventricles. The vascular system has the 5 types of tubes that we mentioned earlier. And the blood consists of 2 components, the plasma and the cells. Students are expected to have a thorough knowledge of the anatomy and the physiology, as well as the mechanics of the heart. With respect to anatomy of flow, this figure is very important. Every single label here has to be learnt, and the order in which they occur with respect to the flow of blood is a common area of testing. So please take your time and somehow learn this. The heart sits inside a cavity, and that cavity has 3 main layers. The endothelial layer is the innermost layer of the heart itself, and that consists of the endothelial cells that are making contact with the chambers and any blood vessels entering and leaving the heart. The next layer is the middle layer, or the myocardium, and that's the muscle layer too. So all the M's, middle myocardium muscle. And that's composed of cardiac muscle cells as the majority, and that's where the muscle contracts to cause the pumping action of the heart. Now outside the myocardium is a thin layer of tissue, a fibrous layer called the epicardium, which has a very close association with the myocardium, and it's hard to separate. Outside this layer is something called the pericardium, which is separated from the epicardium by a very thin layer of fluid. But that's very important because the epicardium and the pericardium are then separated by a cushion that allows the epicardium and the rest of the heart to function, and is lubricated by that fluid. To aid one in remembering the flow of blood, you have to take a journey yourself as though you were floating in a pool of blood, and you can then follow these directions in a cyclical fashion. As just mentioned, the myocardium is actually the muscle cells of the heart, and they are arranged in many layers that are tightly bound to each other, encircling all the chambers of the heart. The heart is in fact a fantastic pump that has the ability to adapt to different circumstances. In addition, its mechanical activity, its mechanical action is evolved to pump from the top down and squeeze the contents in a very systematic fashion. Thus, cooperation between different chambers and different cells, the electrical components, the mechanical components, the hormonal components, the nervous components, all these have to be perfectly aligned in order for the heart to be efficient. When certain cells, the 1% of the conducting system, when certain cells of that system are activated, they trigger an immediate electrical wave that travels through the heart, activating every single cardiac cell. We will talk about the conducting system shortly, but it's important to understand the location of gap junctions between neighboring cells. They accelerate the passage of signals, electrical signals, between cells, and therefore give the heart the capacity to beat in that coordinated and quick fashion when needed. We just spoke about the innervation of the heart through the nervous system. Those nerves are broken into two categories, the sympathetic and the parasympathetic nerve fibers. The sympathetic nervous system is activated during fight or flight, so when you are in a panic, and the purpose there is to divert all the energy of the body into driving the physiology to save one's life. So fight or flight would increase the heart rate, the heart pressure, and the beating of the heart. So naturally, the sympathetic nervous system is part of that network, and those nerves innervate many parts of the heart, and they increase heart rate. And the most common constituent that's released is norepinephrine, a neurotransmitter used by the nervous system. In addition, there is a nerve fiber that innervates the atria only, and that's part of the parasympathetic nervous system, and that uses acetylcholine as its neurotransmitter. The chapter on the endocrine system deals with the different classes of receptors and nerve cells. All we need to understand is that the parasympathetic nervous system has a small set of cells of the muscarinic type, and the sympathetic nervous system impinges on cells that are beta adrenergic. Incidentally, the heart is also responsive to hormones traveling in the blood, and that would be epinephrine. Amazingly, even though the heart is constantly full of blood, it cannot obtain its nutrients or eliminate its waste into that blood. As with other organs, it has to have its own private supply of blood, and that blood originates in arteries that branch from the aorta as soon as the aorta leaves the heart. And those are familiar to us as coronary arteries, and they are the ones that cause problems when they get blocked. The exact location of this branch is the aortic valve cusps. Blood is then pooled together and collected into veins, and the cardiac veins, they drain into a single large vein which empties not into the vena cava but into a coronary sinus at the right atrium. Turning our attention now to the conducting system of the heart, we can see in yellow the cells that are dedicated to moving electrical impulses throughout the heart. The important thing is that the top half of the heart and the bottom half of the heart are separated by a non-conducting region between the atria and the ventricles, apart from a single set of fibers known as the bundle of His, which travel from the top to the bottom of the heart. Reference to this figure is very important over the next few slides. The main electrical excitation region of the heart is the SA node, the sinoatrial node. So back to the figure here, and you can see the sinoatrial node is right here at the very top of the patient's right atrium. These pacemaker cells are very, very important. They generally set the rhythm of the heart at a rate suitable to the physiology of the body. It is these cells which activate the atria to contract. Subsequently, the electrical wave of depolarization then spreads through from the top of the heart to the bottom of the heart through another region called the atrioventricular node. So going back to the figure, we can see that that node is right here. So the pulse of electricity travels from here to here, and in the meantime, the atria at the top are contracting. Once the activity reaches the AV node, then it has a small delay in that communication built in of 1/10 of a second. After that, the information travels through the bundle of His, separating the two levels of the heart, splits into two branches, the right bundle and the left bundle branches, and then spiders out as Purkinje fibers in the walls of the two ventricles. The electrical activity of the cardiac muscle cells, because their numbers are so large, can be monitored from outside the body with electrodes as part of an ECG or EKG. The P wave corresponds to the excitation of the arterial cardiac cells. The QRS wave corresponds to the excitation of the ventricular walls. And finally, the T wave corresponds to the relaxation of the ventricular walls. Unfortunately, the relaxation of the arterial walls is hidden and masked by the massive amount of ventricular tissue. The next section of the chapter can be bypassed and shall be included when we discuss in another video how neurons function. Reiterating what we just mentioned about the electrical conductivity of cardiac muscle, the ECG can be used to monitor the behavior of those cycles. In this figure here, 12.17, this is what a typical ECG from an ideal patient should look like at rest. And we can see the names of the various peaks, the P wave, the QRS system, and the T wave. And in green and orange, we can see what's happening at the membrane potential level with the region of the muscle tissue. One of the most important ways that students can impress is to learn the coordination of the cardiac cycle with all the other activity taking place in and around the heart. This would include the sounds made by the closing of the various valves in the heart that can be heard with a stethoscope, as well as the relationship to the number of beats per minute made by the heart rate and what's happening within the chambers of the heart, and also subsequently how that variation can be manifest to increase heart rate or cardiac output under different conditions. So pretty complex, or is it? Let's begin this analysis by assuming that our heart is beating at 72 beats per minute. That would mean, on a typical heart, that the cardiac cycle will last 0.8 seconds in order to squeeze 72 beats into a 60-second minute. Within that 0.8 of a second, the systole lasts 0.3 seconds and diastole lasts 0.5 seconds. Systole is the contraction of the chambers. Distally is the relaxation or the distension of the heart. So D for D. The heart is a pump. Each chamber is separated from its neighbor by a valve, and those valves exist right here. The first set of valves is between the patient's right atrium and right ventricle, and this is called the tricuspid valve. The next valve is between the patient's left atrium and left ventricle, and that's called the bicuspid valve. The second set of valves also exists on the other side of the ventricles, where the ventricle is connected to a blood vessel through which blood leaves. These are known as the semilunar valves. Reference should be made to figure 12.9 just to clarify these anatomical locations. Unfortunately, multiple other names can also be affixed to these valves, such as the pulmonary valve or the aortic valve. Thus, the heart sounds made correspond to the phases of muscle contraction. This is one of the most important figures in the chapter. Relating the various columns to the various rows is very important in order to get a full picture of how the heart is functioning at rest. Looking at the very bottom, we can see here we have the phases of the cardiac cycle. So if a question asks you about the phases of the cardiac cycle, they're basically asking you about the distally and systole phases. The heart sounds are intimately tied to both these phases. At the beginning of each phase, we have a sound. So systole corresponds to the first heart sound, and distally corresponds to the second heart sound for every time the heart beats. What students have to do now is to equate the sounds and the cardiac cycle with which valves are opening or closing at that particular moment in time. The clue is to remember which sound is made by the closing of which valves. The second major learning objective is to correlate the pressures in the ventricles with the pressures in the arteries during the cardiac cycle. And one of the areas of confusion is this word here, isovolumetric. Iso means the same. Volume means the size of a container. And the metric is a measurement. So isovolumetric would correspond to the same size of ventricle at the beginning of the event and after. Thus, in this context here, we can see how the ventricle can be accumulating pressure without actually reducing its volume. So here the muscles, the muscle fibers are beginning to contract without shortening. They are taking up slack inside the microstructure of each cell. The relationship between the pulmonary artery and the right ventricle is indicated here by pressure changes. As one can see, sometimes the pressure in the artery is lower than the pressure in the ventricles. and sometimes the pressure in the ventricle is lower than the pressure in the artery. And this switchover is important in opening and closing various valves. So step number 2 is where the valves begin to experience an internal pressure to begin to close. And at the end of step number 4, the same thing happens to the other valve. Since each ventricle is surrounded by 2 valves, the first valve to close would be after filling of the ventricle. So the ventricle is beginning to fill, and once it's full, the valve between the ventricle and the atrium begins to close as the pressure builds up. And that makes the first sound. As the blood is then pushed out of the ventricle, the pressure in the ventricle begins to fall. However, the pressure in the artery outside is now greater than the pressure in the ventricle, and that slight backflow of blood is enough to close the second valve between the artery and the ventricle, causing the second sound to be made. Students are encouraged, highly encouraged, to draw this out and to discuss this. Experienced medical personnel listening with a stethoscope are able to listen to the various sounds made by a patient's heart and to make some deductions. These noises, other than the noises expected from the heart sounds, the lub and the dub, are known as heart murmurs. They are caused by defects either in the diameter of the blood vessels or in the valves themselves that are located between the chambers of the heart. This figure here shows you that the normal flow of blood is evolved to be very laminar. The highest speed of blood is in the center, with different layers subsequently slowing down as you approach the walls. However, if the valves are malformed, then you can get turbulence. And the type of turbulence that takes place is an indication of the type of murmur that is caused. The next set of slides deals with cardiac output, the volume of blood ejected by the heart. Cardiac output is measured as the volume of blood pumped out per unit time by each ventricle. There's an equation that we're going to be using quite frequently in the remainder of this section, and that equation is that the cardiac output is equal to the heart rate, a number of beats per minute, times how much blood is ejected from the ventricle per stroke. So that's the stroke volume. So the normal cardiac output, as we discussed earlier, is 5 liters per minute for a resting adult male. Interestingly, if the heart is taken out of the body and all the connections to it are removed in terms of the nerves and the hormonal control, we find that the automatic rate at which the SA node will fire is 100 beats per minute. So an extracted heart will pump at 100 beats per minute. But that's not the normal case. In a normal resting male, the number of beats a minute is about 72. So that tells us that the heart must be outside influence and regulation. The sympathetic and the parasympathetic systems are constantly talking with the heart. Under normal circumstances, the parasympathetic has a greater say, therefore lowering the heart rate from 100 beats per minute down to that 72. Any factor that regulates the heart rate is known as a chronotropic factor. By influencing the molecular biology of the pacemaker cells, one can take a typical frequency of activation, indicated in green, and either slow it down, which is purple, or speed it up, which is the yellow. Again, this would make greater sense once the student has understood the chapter on nerve impulses. In addition to the sympathetic and parasympathetic nervous systems, the plasma concentration of epinephrine can also have a chronotropic effect on heart rate. When we mentioned cardiac output on a previous slide, we were looking at a second component called the stroke volume. The stroke volume is very important. The stroke volume is basically defined as the volume of blood each ventricle ejects during each contraction. The stroke volume itself can be broken down into two parameters. The first is a volume to do with the end-diastolic nature of the ventricle. And the second is the end-systolic volume of the ventricle. This makes sense. So when the heart is dilated, it has a certain volume, and when it contracts, it loses a certain volume. So if you subtract one from the other, the remaining will be the amount of blood that was pushed out of the heart between these two events. The average value is 70 milliliters per beat. Three major factors that can influence the stroke volume are how large the volume of the ventricles is when they are full of blood, i.e. the end-diastolic volume, known as the preload, the amount of sympathetic nervous activity impinging on the heart, and lastly, the pressures, systemic circulation, that push back against the heart, the afterload. All these can be combined together into an analysis first conducted by two scientists, and their system is now known as the Frank-Starling mechanism. Basically, this is a relationship between SV, the stroke volume, and the EDV, the end-diastolic volume. In a nutshell, what this analysis is telling us, this mechanism, is that a normal resting heart is operating mechanically at suboptimum conditions. If you fill the heart to a greater volume with blood, it will pump more forcefully automatically. And if you fill the heart yet further with more blood, then it will pump even more efficiently than it did previously. In a graphical sense, we see this particular profile. So the x-axis has the size of expansion in milliliters of the ventricles, and then the y-axis is the stroke volume generated from this type of stretch of the heart muscle. This data implies that if you stretch the ventricle to about 140 milliliters, then the amount of blood forced out of the heart will be 70 milliliters. If you stretch the ventricle to, say, 210 milliliters, then on the subsequent heartbeat, the amount of blood ejected from the heart will be about 110 milliliters. So the more you stretch the heart, the more it forces out on the very next heartbeat. The conclusion, amazingly, is that within a single heartbeat, the heart can adjust to requirements of the body in terms of pumping capacity. Naturally, there are limits to the size of the ventricle that can accommodate blood, and also a time factor in filling those chambers, because blood coming in from the venous return and from the atria are designed to fill the ventricle. And as the ventricle requires more and more filling, those forces diminish. That explains the leveling off of this curve. But in most cases, we'll be operating down here in this very sort of linear territory. Naturally, there are limits to the size of the ventricle that can accommodate blood, and also a time factor in filling those chambers, because blood coming in from the venous return and from the atria are designed to fill the ventricle. And as the ventricle requires more and more filling, those forces diminish. That explains the leveling off of this curve. But in most cases, we'll be operating down here in this very sort of linear territory. The stroke volume may also be increased by external regulation. The sympathetic neurotransmitter that we mentioned before norepinephrine acts by way of the beta adrenergic receptors to increase contractility of the ventricles. That is the strength of contraction. That is a molecular biology attribute. This means that the ventricles could fill to the same end-diastolic volume, but contract at different rates. Additionally, blood epinephrine can also have a similar effect on the contractility of the heart muscle. The conclusion is that there are external forces that can govern not just the rate of contraction, but also the speed at which these cycles repeat. Examining this data in a graphical sense is what we see in figure 1228. So we have a normal profile here. This is the Frank-Starling curve that we saw earlier. Now, in the presence of sympathetic stimulation, the curve is left shifted. So it shifts to the left and slightly upwards. So under the same level of filling, the force of ejection increases, therefore pumping out more blood from the same end diastolic volume. Graph B shows the change in the ventricles in the presence of stimulation of sympathetic nerves to the heart. So this is the force developed in the ventricles over time in a non-stimulated heart under normal conditions, and this is the same ventricle under sympathetic stimulation. You can see not only does it generate a greater force, but it does so over a short period of time. So this could be somebody who's relaxing, lying in bed, and this will be somebody who will be exercising. And you can see this process automatically increases the heart rate because these cycles happen over a shorter period of time, and that's what an increased heart rate is. But not only that, they eject blood at a higher pressure with each contraction. And that's exactly what we need with a heightened blood pressure when we exercise. The ejection fraction is a measure of the contractility of the heart muscle. It's a simple ratio between the stroke volume and the end diastolic volume. What this essentially measures is the amount of blood pushed out of the ventricle with a contraction related to the size of the end diastolic volume. So whether under hormonal control or not, or intrinsic control, we can measure this ratio, and that tells us how much blood as a fraction of the filling is ejected. And the numbers vary between 50 and 75%. From a physics and mechanical perspective, if the end diastolic volume is the same, but you increase the speed at which the muscle contracts, then the amount of fluid ejected will be a greater amount. Ingeniously, the heart can respond also to pressure outside in the blood vessels. So if the blood vessels are already full of blood and is not being utilized by other organs, then the pressure in the arteries will be great. And that pushback is known as the afterload. The inability of the heart to push more blood into an already full space outside. So that will immediately reduce the stroke volume, therefore preventing further movement of blood into that space. The afterload is very important in regulating the behavior of the heart. Table 12.5 can be used to double check your knowledge. So look at the first column and make predictions on what you think would happen in the two subsequent columns with changes in these parameters. Once you have qualified from the previous table, the next thing you can do is to look at some major factors which are responsible for increasing cardiac output. So the body wants to have more blood circulating from the heart, and we can see how these two mechanisms can both lead to that outcome. So one way is to increase the EDV, the end-diastolic volume, and the second mechanism is to increase the sympathetic nervous system interaction with the heart. This is intrinsic control, this is extrinsic control, outside of the heart and inside of the heart. This system will sense through a roundabout way that the heart needs to modify its behavior in increasing output. But everything relates back to this equation. The output is related to the stroke volume and the heart rate. Patients may have their cardiac functions monitored in two ways. One is through echocardiography, which is a non-invasive technique that uses ultrasound waves to measure the contractions and the state of the valves, and ultimately to measure ejection fraction. And the second, more invasive mechanism is to use cardiac angiography, where a dye is inserted into the patient, and that dye is then allowed to be detected by a real-time monitor as it moves through the heart. The next section is pretty logical, and we can progress through it quite rapidly. This section is called the vascular system. It basically gives you the physiology and some anatomy of the blood vessels that make up the circulatory system other than the heart. So the vascular system has a major function of regulating blood pressure. That is its major function, as well as distributing blood to various tissues depending on the conditions within the body, in addition to external factors also. Anatomically, all blood is surrounded by endothelium. Doesn't matter where it is. It could be inside a capillary or it could be inside the heart. All blood is surrounded by a layer of endothelial cells. Thus, the lowest common denominator are the capillaries, which are basically surrounded by nothing but endothelium. The other types of blood vessels therefore have additional layers beyond the endothelial cells, including smooth muscle, enteric nervous system layers, and connective tissue of various types. The functions of the endothelial cells are many and varied. Not only do they have a physical barrier present between the blood and the tissue, they also act to control the passage of substances such as hormones and local agents. They can also release factors that allow new blood vessels to grow into tissue. They can allow existing vascular systems to be rearranged. They can also contribute to the formation of extracellular matrix by releasing growth factors. They are very important in secreting substances that allow clotting and clot removal. In addition, these cells can manufacture hormones as well as extract and degrade hormones. They can also release local agents that influence the immune system, and a negative role as in atherosclerosis, where these cells can impact smooth muscle growth. Figure 12.32 has on the x-axis a location, and on the y-axis it has pressure in millimeters of mercury from 0 to about 140. And what's obvious is that the systemic circulation has a much higher variation in pressure than does the pulmonary circulation. And as you can see here, there's a drastic drop in the systemic circulation's pressure as you move away from the arteries through the arterioles, and almost zero pressure when you get to the end of the veins and return back to the heart. Let's take a quick journey through the various types of blood vessels. Once the blood leaves the ventricles, it enters arteries. Let's deal with the arteries on the left side of the heart. The very first artery is the aorta, and it's the largest artery in the body. Arteries share certain features in common. The first of which is that they have thick walls. The reason they have thick walls is they contain large quantities of connective tissue with elastic fibers. These elastic tubes allow the artery to recoil once it's stretched by the blood pressure from systole. In addition, on average, the arteries have the largest diameter of all the blood vessels, and this helps them to serve as low resistance tubes for the flow of blood from the heart to the organs. Finally, all arteries to a large degree act as pressure reservoirs. That means that they can absorb and stretch during systole and then rebound and push back and continue to flow of blood when dystole takes place. A term that we learned with the lungs was compliance. Compliance is simply the ability to be pushed into a new shape and be stretched. So the higher the compliance of any structure, the more easily it can be stretched. However, arteries are not as compliant as veins are. Veins can easily be stretched with a little bit of pressure. Arteries need a bit more pressure than veins, but still they're pretty compliant. A simple formula allows us to understand the components of compliance. So compliance of any structure is equal to the change in volume divided by the change in pressure. For most of the length of arteries from the heart, one can measure pressure fluctuations because of the activity of the cardiac cycle. This figure here is for a young adult male, and the pressures average out to be 10 millimeters of mercury higher than in females. Regardless, the profile is the same for males and females. The bottom axis is time, and the y-axis again is pressure, going from 0 to about 140. As one can see, diastolic pressure is the lowest when the heart is filling. But still, it's 18 millimeters of mercury. It's not 0. So this is caused by the rebound that you see here from the walls of the arteries. Maximum pressure in an artery is obtained during systole, when blood has left the heart. Once the ventricle has stopped pushing, there is a sudden drop in pressure, but there's this little notch here, and that's the aortic valve closing, which causes this slight rebound as the valve takes up slack. But as soon as the valve has been stiffened, then the pressure rises slightly again, and then the artery is responsible for continuing to push the blood into the tissues as the heart takes the next beat. The average here is the mean pressure, and we'll talk about the mean pressure in a second. Another component of physiology is age. As the subjects get older, there's a change in the compliance and the pressures that are generated in the arterial system. Young people, they have a mean pressure about 65 to 70 millimeters of mercury, where somebody in their 70s and 80s will have a mean pressure close to 100 millimeters of mercury. This is the blood pressure, so systolic versus diastolic, 150 over 70. Pulse pressure is simply the difference between the systolic blood pressure and the diastolic blood pressure. For instance, if the systolic is 120, diastolic is 80, just subtract the diastolic from the systolic and you end up with 40 millimeters of mercury. So that's the pulse pressure. Now, the pulse pressure can be felt in localities throughout the body as a pulsation or throb. Important parameters contributing to the size of the pulse pressure are the stroke volume, the amount of blood leaving per beat, the speed of ejection of the stroke volume, and arterial compliance. A disease called arteriosclerosis is a stiffening of the walls of the arteries, and naturally, because the arteries are now stiffer, they're less flexible and less likely to obey pressure changes, so they have a reduced compliance. The next commodity to learn is MAP, or mean arterial pressure, and the way that's calculated is straightforward. You take the diastolic pressure and you add to it 1/3 of the pulse pressure. Remember, the pulse pressure is the difference between systolic pressure and diastolic pressure. Why is this number important? Because the MAP allows you to determine the average pressure driving blood into tissues because of the entire cardiac cycle. Sphygmomanometers, or blood pressure cuffs, allow you to measure blood pressure, and the relationship between blood pressure and these devices is illustrated in this figure. The stethoscope has to be placed on the far side of the cuff with respect to the position of the heart. The cuff is then pumped up until it's higher than all the blood pressure, therefore cutting off blood flow to that limb. As the pressure in the cuff is reduced, when you reach the higher blood pressure value, the systolic reading, then and only then you'll hear sounds as blood is able to push its way through into this region of that limb. Then the sounds will get louder and start producing various sounds until you reach a point where the pressure has dropped so much that blood flow in the vessels becomes laminar again once it hits the lower value for blood pressure, the diastolic. And at that point, no further sounds will be made because blood flow is silent. The smaller arteries, known as arterioles, are slightly different from their cousins, the arteries. They have two major physiological functions. The first is they are responsible for delivering blood to organs based on the organ's requirements. The arterioles are more important than the arteries when it comes to determining MAP, the mean arterial pressure, because all blood has to flow through arterioles before it reaches all the different organs of the body. The diameter of these arterioles is controlled by three sources, including the nervous system, the endocrine system, and of course local mediators of physiology. A simple variation in the diameter of these blood vessels is required to either increase or decrease blood flow. This lab equipment does a fantastic job of giving you an analogy with respect to how arterioles work. So here's your blood in the arteries, and the arterioles are then diverted to various organs, and they have smooth muscle along their lengths that's controllable. So in this scenario here, blood flow is greatest to organ 2 and 4, with the least amount going to organ system 3. In an alternative sequence of physiological conditions, the body is able to readjust the openings of these arterioles and therefore divert blood to other organs and organ systems. The arterioles closely follow the flow-pressure relationship that we've seen before, where the rate of flow is proportional to the pressure difference across that region divided by the resistance. In simple terms, you can increase the flow either by increasing the pressure difference or reducing the resistance. These arterioles are under a form of local control. Local control is one of the takeaway messages from physiology. The central nervous system does not have enough capacity to monitor and control every aspect of your physiology. Therefore, much of your physiology relies on local decision-making made at the level of cells and tissues, and to a large degree also including organs. This local control is illustrated here with two examples. For instance, if the metabolic activity of an organ increases, that leads to the consumption of oxygen, therefore the amount of oxygen decreases, and the waste products, the metabolites, are increased, and that's then referred over to the interstitial fluid surrounding those cells. The arterioles dilate in response to these substances or to the absence of oxygen, and because they dilate, they then are able to redivert more blood to that organ. In the lower example, we see autoregulation, where the flow to the organ is fluctuated around a set value. If the organ begins to experience a decrease in the arterial pressure, that's then translated into a decreased flow into the organ. Oxygen levels drop, metabolites increase, and in this case there's a reduced stretch of the walls of the blood vessels entering that organ. That is then translated into a dilation of the arterioles, and that restores blood flow very quickly. So this is a very dynamic system. It's like driving down the highway and using your accelerator to match a speed that you wish to drive at without using the cruise control. Flow autoregulation may be one of the best takeaways from this section, in that it corresponds nicely to the automatic adjustments that can be made to blood flow in proportion to the needs of that tissue at any instant in time. Local metabolic factors alter the diameter of the local arterioles, and that is known as myogenic control of arteriolar diameter. So these metabolites, they influence the muscles that are encircling the arterioles at that location. In this example, we're looking at skeletal muscle and the blood supply into them. Vasoconstriction and vasodilation of skeletal muscle arterioles can be accomplished by changing the diameter of the smooth muscles in the arterioles feeding those locations. So epinephrine causes an effect on both, whereas sympathetic postganglionic neurons are able to cause vasoconstriction. Endothelial cells themselves are also responsible for producing and secreting paracrine agents that can diffuse into the adjacent tissues and influence smooth muscle behavior. And these agents can either relax or constrict these muscles. One of the most important ones discovered in the last 10 years or so is nitric oxide. Nitric oxide is a relaxant, a vasodilator. So once nitric oxide is produced, it can cause the increase in blood flow to that particular organ. This is how Viagra works in erectile dysfunction. Amazingly, many factors are being discovered or are already known that can have a localized effect on arterial radii, in addition to neural and hormonal controls. Capillaries are fascinating in themselves. Even though they may consist of a single layer of epithelial cells, they are still very complex in their physiology. Capillaries are found all over the body, with one exception, and that's the cornea of the eye, which needs to be clear in order to see the world. The primary function of capillaries is to remove waste products, deliver nutrients, and also exchange gases with the surrounding interstitial fluid through which they pass. They accomplish this by simple chemical diffusion. Here is a cross-section of a typical capillary. You can see it's just wide enough to accommodate a red blood cell or two, and in this way the vicinity of the tissue is brought into close proximity to the red blood cells themselves. Histology reveals that there's a microcirculation present in various locations in all tissues, where the arteriole and the venule are separated by capillaries. And at the surface of the branches from the arteriole to the capillaries, we have these pre-capillary sphincters or muscle bands that can respond to local conditions in this region of this tissue. So the microcirculation is even finer control over the amount of blood passing at a very, very local level within a system. The capillaries may be thin, but there's so many of them that in fact 5% of the total body's blood at any one moment is localized within the capillaries. Once that fact is understood, then this chart here becomes more discernible. The red line is the cross-sectional surface area of those particular blood vessels, and again, capillaries are the largest. The green line is the velocity of blood taken as a whole passing through those localities. So blood flows the slowest through capillary beds before picking up speed as it merges into venules and veins. The pressure in the blood capillaries is enough to cause filtration. Filtration is also known as bulk flow, and that is sufficient to deliver to the interstitial fluid and the cells bathed in it the nutrients and substances that they need to continue to survive. At the same time, absorption of waste products and carbon dioxide takes place again back into the blood. The movement of materials across capillaries is governed by both the physical forces and the chemical nature of the components present in that environment. The net movement is dependent on a number of other parameters. The first is hydrostatic pressure. That's the force exerted by the blood pressing against the walls of the capillaries. That's blood pressure. The second component is that in the capillaries, the pressure tends to force out, through filtration, bulk flow material, especially at the arterial end, where because of the arterioles, pressure is the higher of the two. As we remember from a previous diagram, as you migrate across a capillary, the pressure begins to decrease. The third component is colloid osmotic pressure, oncotic pressure, and that's the force that opposes the blood pressure, the hydrostatic pressure, and this is pressure caused by the proteins present in the blood pulling back on the water component in the interstitial fluid. This pressure is more or less constant. It doesn't matter where you are within that capillary. In order to calculate the overall effect, which is known as the net filtration pressure, NFP, we have to marry all the components arithmetically together. So PC is the blood pressure, the hydrostatic pressure. PF is the hydrostatic pressure of the interstitial fluid pushing back. And pi C is the osmotic pressure of blood due to these blood proteins. And pi F, or IF, is the osmotic pressure due to the dissolved components of the interstitial fluid, mainly its proteins. This slide here gives you an example. On one side of a capillary, and then on the other side of a capillary. The values are taken from these mentioned just above. You can see here the filtration pressure is 10 millimeters of mercury. That means there's a net force pushing fluid out of the capillary in this case. But a short distance away at the other end of the capillary tube, the reverse is true. There the pressure, net pressure, is to allow some of the fluid at least to re-enter the capillary. Now the pressure difference between the two is exactly the same. So here the filtration pressure is 10 millimeters of mercury. Here the absorption pressure is 10 millimeters of mercury. The amazing thing is that this little seesaw of forces is enough to allow the capillaries in the body to generate a net outward force that's slightly greater than the pulling force back into the capillaries. So overall there's a net filtration of about 4 liters a day of intracellular fluid being generated. And that fluid cannot accumulate in the tissues, otherwise you get swelling such as edema. The fluid normally makes its way through the lymphatic system, of which we shall be talking about later, and that returns that fluid through a complex series of canals back into the venous circulation. The combined actions of all these control elements is so powerful that for a particular organ, the localized controls can vary the amount of blood and the blood pressure associated with it entering the organ itself. So, for instance, the orange line is the initial state at which that organ may exist. Then with vasodilation, there's an increased flow of fluid into that tissue. And with vasoconstriction, the purple line, there's a decreased flow, and in such that you can also cut off the flow altogether if you wish. The values on this slide simply indicate that 60% of the blood volume is present in veins, the low pressure side of the circulation. Talking about veins, let's now concentrate on veins and venules. The thing they share in common is that their walls are in general thinner than corresponding arteries. They have less smooth muscle, and they also are much more compliant. Therefore, it doesn't take much blood to stretch them in terms of blood pressure. In addition, veins have something called capacitance. Just like a capacitor can hold on to electricity, veins can hold on to blood. So they become a reservoir of any extra blood that may be wandering around the system once it leaves the organs. The relatively slow flow of blood through veins compared to arteries is also important when it comes to the immune system and the migration of white blood cells into local tissue, a process called extravasation. The average blood pressure in veins is about 15 millimeters of mercury. To encourage this blood to keep moving back towards the heart, the body has evolved 2 important mechanisms. One is the negative pressure manufactured by the breathing apparatus in the thorax. So this is known as the respiratory pump. So as you breathe, there's a sucking effect on your abdomen and other parts of your body, which tends to draw blood towards the heart. The other mechanism is more universal. These are called muscular pumps. So whenever your muscles squeeze on the veins or venules, they cause the movement of blood. Because veins have within them valves, trapdoors, that work one way, the blood is encouraged to flow towards the heart. As an added measure, the sympathetic nervous system also impinges on the smooth muscles lining the veins, and when activated, that causes contraction and reduction in the diameter of the veins, forcing blood back towards the heart. Figure 48 illustrates both the nature of the muscle pump and the presence of valves in the veins, combining the three parameters that we just discussed, namely sympathetic activity, increased muscular pump effectiveness, and the inspiratory sucking action of the respiratory system. Combining that with what we learned earlier, that blood volume can impact venous return. All these are responsible in a knock-on effect in regulating the stroke volume. Let's break the video here because I'm having trouble with my computer memory, and then I'll generate a second part for the circulatory system and the heart. Thank you.